All right, thank you so much. And I also want to express my thanks to the organizers for including me and my regrets for not being able to be present here much. This is our last week of classes. There's 2 faculty meetings. It's a tough week, but I would love to be spending more time with you. So if you have questions about this material, please email me after. I love engaging by email after talks, and will absolutely respond to you, and we can carry on the conversation that way. Becky, can I make a request for a bottle of water?
Yes.
Thanks. Okay. I'm going to be telling you about a project we have going in the lab for bringing together Bayesian methods, which are highly principled approaches to characterize uncertainty in our inference about biological systems, and dynamical models of connectomes, which are simulations of brains. And we're doing this in the context of understanding individual behavior, which is, oh, thank you so much. Great. Which is what our lab studies. And can you hear me in the back okay? Yes, great. So these images here kind of depict the variety of techniques we use in my lab. These include tracking of behavior of individual animals. So you can imagine this fly walking up this stick, and it's got to make a choice about following the left or the right branch. We use computational methods, and so depicted here is a raster plot of a simulation of the olfactory system of the fly. So every row here, and there's 3,000 of them, represents a neuron in the simulation, and time goes across the x-axis, and each of those vertical stripes represents the presentation of a different fictive odor to this simulated brain, and we're getting patterns of activity across that network. And we also use molecular and circuit methods to establish causal relationships between variation in individual circuits and variation in behavior for those animals. And I have been told that this is a diverse audience and that you should all be very free to jump in with questions, even if they're basic. So feel free to ask me about the biology or the math or whatever you like. Okay, this is another framework we bring to this question. We're particularly interested in explaining behavioral differences between individual animals that have the same genome and have been raised in the same environment, right? So this framework kind of challenges the canonical wisdom that variation in phenotypes comes from variation in nature and nurture, nature being genetic differences between individuals and nurture being environmental differences between animals. In this case, we can make their genomes the same, and we can grow them in standardized lab environments. And as I'll show you, their behavior is still very different from each other. And so the question is why? Because in principle, we've matched nature and matched nurture. But the general idea we have is that the sort of inexorable fluctuations of being a system down to the sort of thermodynamic scale propagate upward through development and through the sort of operating physiology of the organism to produce variation at the macro scale. So in this diagram, we have depictions of phenomena at different scales, spatial scales, and the cartoon, there's a grayed out kind of silhouette for each cartoon, which represents alternative outcomes to what you're seeing at each of those scales. So here's like water molecules and they're vibrating, but maybe they vibrated a little differently in one animal compared to the next. And presumably that's true, right? Like the sort of detailed thermodynamic variation is going to be completely different from one animal to the next. And maybe that ends up propagating to the scale of, in this case, transcription. So this is the expression of genes, the conversion of the information in DNA into RNA, and it's done by protein enzymes. And you can imagine with those water molecules bouncing around in different ways, the residence time that that enzyme has on the DNA might be different from one pass to the next, resulting in different numbers of transcripts of genes, which results in different numbers of the proteins that are encoded by those genes being expressed on the cell surface, leading, for example, to different migrations of the growing neuron as it tries to find its synaptic partners, leading to different integration of individual neurons into circuits, different wiring patterns that biases the animal to show different patterns of behavior, right? Now, this is all a hypothetical framework, right? right? And what's challenging about this work is there aren't really tools to manipulate the sort of thermodynamic churn that sits at the bottom of this mechanistic hierarchy. We have tools that let us manipulate circuits and manipulate genes, but they're limited. Also, there's a veil of stochasticity where the events down here may be sort of fundamentally deterministic, but we can't see them all. And it's probably fundamentally impossible to measure all of these things. And so in some sense, there's an opacity here, which we might as well treat as a random factor. So we think of like what we're studying are how nanoscale and microscale random fluctuations propagate upward through layers of causal regulation to produce macroscale differences in behavior across individuals. Okay, so I'm going to tell you today about work that started in studies of the olfactory system. And when we get to the point of simulating brains, we'll be focused on simulating the olfactory part of the brain. And this was one of these examples of flies have different individual behavioral patterns. You could call these personalities. They persist for many days or weeks, essentially the lifetime of the animal. If you want a human equivalent, it could be like, well, what did you choose at the breakfast buffet? Did you take the quiche or did you take the fruit, right? Maybe that reflects kind of a typical pattern you have of, I like sweet things over savory things, right? This is the sort of equivalent behavior that humans exhibit to what we're looking at in the flies here. So I'm going to play this movie, and what you're seeing is a top-down view of flies walking in these linear chambers. And there's invisible tubing delivering flows of odorants into these chambers. And those, when the odors come on, it'll be marked by a pink bar and a green bar here. And odors flow in both sides of these chambers and are evacuated out the middle. And the flow rates are set up so that this is laminar flow. So what you get is kind of an even filling of odor A from one side and odor B from the other, and that they meet in the middle, and there's a relatively sharp boundary because there's not a lot of mixing before the odors get pulled out of the chamber. So it's kind of like a sort of two-zone assay, and we can extract individual odor preference from this by just tracking the flies and seeing where do they like to hang out. Do they hang out on the pink odor side or the green odor side? The specific odors are these monomolecular alcohols, 3-octanol and methylcyclohexanol, and they smell a bit like stink bugs and kind of like whiskey / dirty socks is the other one. But these are aversive odors. So if you want the human version of this assay, it's not quiche versus like croissant. It's like, do I hang out in the room that smells like skunk, or do I hang out in the room that smells like garbage, right? And we probably, so raise your hand if you'd hang out in the skunk room. Okay, raise your hand if you'd hang out in the garbage room. Okay, so like we did pretty good at like splitting 50-50. And some of you who didn't raise your hand, maybe you're kind of ambivalent. Those are both bad choices. So you'd split your time. And what we see is this like broad unimodal distribution across individuals, where some spend all their time in octanol and some spend all their time in methylcyclohexanol. And the most common kind of fly is about 50-50. These are like the standard odors that have been studied for about like 40 years in the context of olfactory learning in flies. So it's really just kind of adopting standard odors. Monomoleculars tend to be aversive. You need sort of more complex, richer mixtures for them to be appetitive. There certainly are appetitive odors we can deliver in this rig, like apple cider vinegar smell or like yeast volatiles. But just for sort of keeping things simple and tracking precedent, we went with these. Yeah, okay. So the beginning odors are gonna be off. You won't see those bars. Then the bars will come on. That's when the odors are being delivered. And I recommend tracking this like lime green fly and this Kelly green fly here.
It's the same pair both.
Yeah, everybody's getting octanol on the top and MCH on the bottom, yeah. And then the odors go off, and you can see now they sort of stopped caring about that boundary. So did folks appreciate that the lime fly spent all its time on this half and the Kelly green fly spent all its time on that half? So that's like kind of your anecdata to show you that individual flies have distinct odor preferences. Okay, the lab is interested in sort of a couple major forms of questions about individuality. One of which is what controls the extent of variation? So what genetic elements and neural circuit elements control variance as a trait? This is like saying if I'm rolling dice animal by animal to establish a behavioral preference, how many sides are on the die? Like what's the range of outcomes that could be achieved through that process? Another question we ask is what accounts for individual outcomes? Why did this animal prefer octanol? Or why did this animal prefer methylcyclohexanol? And that's like saying, when I roll the dice, why did I get a 7? So this question is in that second category about what is accounting for individual outcomes. And the sort of term we've come up with is we're looking for loci of individuality. These are the places that in the neural circuit there is some variation across individuals that accounts for the difference in their behavioral biases. And just to sort of paint a very coarse conceptual model for this, imagine the brain as a like feed-forward sensory motor transformation process. We know brains are not this, but just for the sake of the argument, transforming stimulus information into motor outputs. And in this diagram, yellow indicates identity across individuals, and blue indicates idiosyncrasy across individuals, right? So we give the animals the same stimulus, but then we look at their behavior and they are behaving differently from each other. So at some point, there's some transformation of the representation of the information from being identical across animals to being distinctive across animals. And presumably, like if you look at the neural layers immediately preceding motor output, you could decode the individual because like those are the signals that are controlling the leg movements that we are reading out in the behavior. So presumably by the time you get to that like second to last layer, the signal looks like the idiosyncratic behavior. But maybe if you measure out in the peripheral olfactory system, maybe all the animals are reading information about the odor environment in the same way. Yeah.
I'm sure you rolled for this, but I was just wondering, sorry, I'm still back on the last slide. If an animal starts sort of at, let's say, the bottom edge of this,
yes,
then they're more likely to have habituated by the time they get to the other stimulus. Is it possible they just smell it and turn around and then they just stay in the one they're habituated?
There is a slight kind of like 10% of the variance neophobia response. And let's just like play the movie again and you'll probably see examples of this, where as soon as the odors come on, most of the animals that when they encounter that boundary the first time, they turn around. And then they tend to stay in that compartment where they turned around. But that's like sort of 10% of the variance of the preference signal that we get.
I see.
Yeah. And so if you like measure the preference that the animals have on day 1 or on Monday, let's say, and then measure them again on Friday, you'll see a certain correlation. And part of the reason that that correlation isn't perfect is like these sorts of effects. Yeah. Just for some context on the experiments, one of the reasons this is like a challenging and frustrating modality to work on is that these animals habituate very quickly to these odors. And maybe part of this, Ofer, is that like these are not naturalistic odors. Like they know these are weird, and so at some point they can learn to ignore it or ignore it readily. And so we actually kind of get like a 3-minute window where they're sort of, effectively responsive to this choice. And so it's a limited number of choice events per animal per experiment. It's sort of on the order of 20. I showed you that fly walking on the Y-shaped stick. So another thing we study is the individual tendency of animals to turn left or turn right. And we just put them in Y mazes and let them walk around for hours. And they'll generate like hundreds or thousands of choices. So there's a lot more data for some measures of behavior than others. And olfaction's kind of on the low end of the scale of like how much we can measure per individual, which adds some noise to the statistics, just for context. Okay, so we have this framework of transforming potentially identically encoded stimulus information into idiosyncratic motor outputs, and the loci of individuality are the places in the circuit where that transformation happens. So in this cartoon, it happens at this synapse from like the third neuron into the fourth neuron, suddenly it looks like an idiosyncratic response. Of course, you can imagine that maybe there's developmental noise across all the synapses in the brain, and so you get this kind of gradual process of converting that signal into something idiosyncratic. And what's lovely about flies is we know how its brain is built, and we can potentially probe these levels. Like we can take the idea of the cartoon seriously and get genetic access to the successive layers of the olfactory system and potentially like color code these dots by how much the encoding of information in them reflects the identical input or the idiosyncratic motor output. And I sort of won't belabor the details of the kinds of neurons because I want to get to the models, but if you're an aficionado, the olfactory system of the fly is very similar to the olfactory system of the mammal in terms of its sort of topological organization. And particularly interesting neurons are the olfactory receptor neurons that bind to odorants and transmit olfactory information into the brain. They do this in discrete channels that are represented morphologically in features called glomeruli in the antenna lobe. And then within each glomeruli, there's 2 to 4 projection neurons that take the total activity in that glomerulus and then like project it forward into the brain to association centers and centers that drive innate behavioral responses. So projection neurons and olfactory neurons, they both have projections into this glomerular format. So like within the antenna lobe, there are these 50 channels, and they contain the axons of olfactory receptor neurons, the outputs of those neurons, and the dendrites of projection neurons, the inputs to those neurons. This is super convenient because we can record activity across the antenna lobe, so potentially looking at a large number of those glomeruli by expressing neural activity indicators either in olfactory receptor neurons or projection neurons. But what we end up is with like a vector that represents the activation of the olfactory system. And that vector has the same dimensionality, whether or not we're recording out of ORNs and PNs. So it makes kind of the modeling analysis very convenient because we can compare the representation of information at the sensory periphery or one level deeper into the brain in the same mathematical format, in the same vector representation. So it makes the kind of head-to-head comparison of saying, does activity in the projection neurons look more like individual preference compared to ORNs, and if so, by how much? Because those vectors are in the same format, we can potentially do that comparison quantitatively. So here's the experiment. This was work done by Matt Churgin and Danilo Lavrentovich in the lab and published last year in eLife. And I've shown you already the behavior rig. So this is that camera that recorded the top-down view of those linear arenas, which are here. And then this is plumbed with tubing that provides those monomolecular odors when we want and with the right dilutions. And then the camera tracks the behavior. And so we get this kind of abstracted readout where there's one odor compartment and another odor compartment. We track where the flies are. And the individual preference measure is the fraction of time they spend in one of those compartments, just a reference compartment. And what's coming is an experiment where, oh, Anne, yes.
Just about your framework we were given, I'm wondering if the site of the individual preference has to be the same for each choice of each individual. I'm thinking like if I gave you 10 and one has cilantro in it, then a certain subset of people will just like rule that out right off the bat. So does it always happen in the same place?
Are you asking, does it always happen in the same place within a modality, like within olfaction or something?
Within a particular pair of choices.
I will say we don't have, this is not designed in a way where we could tell you if it was happening in different places in different individuals, right? We're going to be looking for the statistical association of variation in the activation of glomeruli and an individual's odor response. So the measure is a correlation where the data points are individuals. So I don't think it's like set up to detect differences in that. We're either going to like find that it's predictive or not, or to what extent. I think that's totally conceivable, and this isn't really something I thought of before, but that's super interesting. As for different odor pairs, I think conceivably we could detect that. These are challenging experiments because it's like measure behavior in a finicky assay and then do calcium imaging and do this on a scale where data points are individual animals. So like we can't get away with like, N of 8. Like we end up sort of going for N of 50 or 60 calcium recordings per correlation we're trying to measure, trying to get to that statistical scale on the activity data. So we haven't found ourselves with the throughput to like really probe a whole bunch of odor choices. The paper does have, so it has octanol versus methylcyclohexanol, and it also has octanol versus air. So we did find that the signal of idiosyncrasy sits in two different places in those two different comparisons. For odor versus air, it's more peripheral and may have to do with sensitivity. And for odor versus odor, it's like one step down, where maybe we're getting lateral comparisons between the representations of these odors. Yeah.
In the middle of the platform, there is this suction motion.
So there's a negative pressure,
Yes.
like at the side, yeah.
So is that always on during send? Or
It's on through the whole thing. What we change, so like in the pre-odor period, there's flow of clean air forming that sharp boundary, and then we just like inject the odors, and that's what you saw.
but not turned on only for the smells.
The flow is not only on for the smells. There's a little like clicking of the valves, the solenoid valves, but other than that, it should be identical. Yeah. All right. So the experiment is going to be to measure individual odor preference in the style of the movie you saw, and then take those flies and mount them for activity imaging. We didn't do any real-time or the concurrent behavior measure at this point, so the flies are literally just dangling under the stage. Their head is windowed, and we're measuring neural activity in the brain with calcium indicators. And then we can flow odors onto the fly and see how the antenna lobe responds. What we collect is this matrix of responses across odors and glomeruli. There are 5 different glomeruli. I promised 50. We didn't get anywhere close to that for technical reasons. We got 5. We're reasonably happy with that anyway. And 12 odors, so 60 measures of odor glomeruli responses, which we turn into a 60x1 vector, which is now collected across individuals. Right, so each row here is a fly, and each column is an odor glomerulus pair, and the color indicates how active the neurons were for that combination of glomeruli. So that's odor channels and odors. And then we want to ask, do the values in the 60x1 vector predict the single value, which is odor preference over there? Now, obviously, you could take like your favorite model, linear model, whatever you want, and predict one number from 60 numbers. And this is another reason we're trying to get throughput, is to like have some flexibility in the models without totally like having way more model parameters than data points. We took a conservative approach, which we're going to take this variation in these 60 values across individuals and do principal components analysis on it, and generate, and just look at like the first 5 principal components and ask, does each of those 5 principal components correlate linearly with our behavior measure? So we found that the second principal component, so it's small, I'm sorry about that, PC2 there, has a significant correlation with the observed odor preference when we look in projection neurons. So by the time we are measuring activity in that second order neuron, there are signatures of idiosyncrasy in how they respond to odors, which are predictive of the behaviors that the animals exhibited previously in the odor choice assay. This correlation is absent if you look one step upstream in the olfactory receptor neurons. And so here is a depiction of the loadings of the principal components. So this is how much each of these 60 odor glomeruli pairs contributes to these dimensions of variation in odor response, and highlighted is PC2. And what you can see, I saw your hand, I'll get you in a second, is that the sort of colors of these loadings, which indicate positive or negative loadings, cluster by these DC2, DL5. These are the glomeruli. So what we're seeing is that the organization of the variation of odor responses across individuals goes by glomeruli. So regardless of what odor was delivered, in this case, like an individual might have a strong response in DC2, whatever odor it is, and that would be anti-correlated to their DM2 response, whatever odor it is. Okay, what was your question?
Correlation being absent in the presynaptic foreign activity.
Yeah.
So you may have just asked this after I hand up or answer this, but is that like you checked all the first handful of PCs, or you check these glomeruli in particular? Like, how can you sort of rule that out?
We applied the same framework, which was like, let's collect this individuals by odor glomeruli pair matrix, do PCA on that, take the first 5 principal components, and in those 5, we didn't see any that were significantly associated.
And then when you kind of, in a supervised way.
Oh, yes, that's true for this. Okay, I'll elaborate, but go ahead. Yeah.
No, just like, then I guess you could check the presynaptic activity at these glomeruli themselves, but maybe you're going to get that.
It was the same glomeruli for both. And that's drawing on this like convenience of the ORN axons and the PN dendrites are segregated spatially in the same way. So we can represent their vectors identically for the purposes of the modeling. Yeah. So the thing I was going to elaborate on is in the odor versus air analysis, PC1 of the ORNs is significant for the individual preference. And PC1, the loadings of it, you can see sort of.
Getting lost in the acronyms.
Yes.
ORN again.
ORN is olfactory receptor neuron. That's the most peripheral neuron that binds to odorants. So it's in those neurons where we see that PC1 correlates with odor versus air response. And the PC1 loadings of ORNs looks like the PC1 loadings of PNs. And you can see it's like pink or red through all of them. So this is like a measure of sensitivity. And so the sensitivity of ORNs predicts individual variation in odor versus air choice. And that makes sense. If an animal smells the odor more easily, it may be more averse, may be more repulsed by this aversive odor than if it is less sensitive to the odor. Kristen?
Possible to look at the time to see how static this is for a single time?
Yeah, technically. So there are folks who have done chronic recordings in flies, and this involves like using epoxy and glass to like seal up that dissection patch that you make in them. There's a few labs that have managed to do this. We haven't tried.
But even maybe even over like that three-minute period that you were talking about, when they get desensitized to the odors.
Yeah. So there's a student, Madison, in the lab working on getting a ball under there so we can measure odor preference in real time while we do this. You can look, so I didn't really explain the details of the imaging, but it's like that panel of 12 odors and then repeat it as many times as the fly will take it before it dies. And so like for some flies, we'll get like up to an hour with multiple repetitions of the odor panel. And the idiosyncratic calcium responses are consistent across that hour. So we're not day to day on the activity, but hour to hour.
So Ben,
Yeah.
for clarity, could you go to the drawing that you had of the thing glomerular? So this may be too technical. So in the glomerular layer, so what you're calling glomerular response is the dendrites of the principal neurons.
Of the projection neurons. Yeah.
Those are labeled by how? Because you have GCAMs and those specifically?
Yeah, we can like do a post-synaptic PEG GCAM.
Over sense label. So,
Yeah.
so to answer to Juan Carlos' question, you just did the same in 2 different labels and
Yeah.
Okay.
2 different batches of experiments.
Make it very clear.
Yeah. Yes.
Individual by odor glomeruli matrix, would you describe that there are high response individuals in general and low response individuals in general, versus it's just a matter of odor preference, like everyone has roughly the same baseline?
When I squint at this, what I see is like, I can see the columns, right? And so the columns represent the stereotypy of odor responses across individuals. And I can also see that like some rows are brighter than others. It's like barcodes in two dimensions. And the brighter rows and dimmer rows is what you're saying, more sensitive or less sensitive individuals. There's some question about sort of technical noise there, because it's like if you didn't do as good of a dissection and there's more like schmutz sitting on top, maybe you get a dimmer signal out of every glomerulus. But on the other hand, we can predict the behavior that we measured separately from that variation. So it's probably real. But I think that's my takeaway, is like sort of broad stereotypy, as you said, some variation individually in the sensitivity. And that's why sensitivity shows up as PC1. And then the kind of differences of like DC2 versus DM2, like I don't really see that just looking at the matrix, but that's what principal component 2 is. And then principal component 3 is kind of DC2 versus everybody else, something like that. Yeah, yeah.
Are these behavioral preferences heritable? Like if you cross?
They are not. Yeah.
Like in the odor learning field, right, there's always this like innate preference thing that they see to begin with, and then they can overwrite it with learning.
Yeah.
How does this relate to learning?
What I can tell you there is that if we silence the associate, so there's sort of, we don't know to what extent the circuitry which provides a substrate for associative conditioning is just like operating all the time. Or is it really only there when there are these kind of like discrete salient events? Like when we train flies, we give them an odor and we give them an electric shock and they freak out, right? And things like that don't really happen in the vial, but they do get in fights in the vial, or like maybe go through periods of starvation. So it's unknown. But if you look at the sort of variance of preference across individuals and then ask, to what extent does that variance change if we silence neurons associated with olfactory associative conditioning? The answer is it doesn't change. As if, as if the learning isn't really part of the story, but it's not definitive. Yeah. Yes.
Yeah, basically, I think in this field have several image of solution. One is connectivity, synaptic, synaptic based image. The other one will diffuse
Yes.
scenery.
Yeah.
The third one will be electrophysiology.
Yeah.
Have you write any equations that integrate these three things?
I will show you equations if we get there. Should I jump ahead or should I just?
It's up to you.
Okay. I'm gonna skip this. There's other correlates. All right. When we got to this point, and thinking back to the sort of grand conceptual framework about variation at the nanoscale propagating up to the macro scale, how do we test like causal relationships? Like what we have coming out of here is a correlation where there seems to be some difference in responses that correlates to preference. But how do we show that this is one, causal, and that two, the variation we see in odor responses is due to that sort of nanoscale developmental fluctuation versus some other explanation? And there aren't tools to turn off developmental variation the way there are tools to turn on and off neurons in flies. So this is like one of these challenges of studying variation and individuality. So how do we test the hypothesis that developmental stochasticity in the ORN to PN synapses causes different behavior and different calcium responses? So this is where we turned to computational models of the brain, because this is a digital sandbox where we can do things like include developmental variation or exclude it and ask, how does that affect the variation in odor responses of our simulated brains? So we are using, it's a rudimentary model of a neuron. It's a leaky, integrate, and fire model, which means that the neurons have, we're tracking the voltage of every neuron. And when upstream neurons fire, downstream neurons pick up some voltage, and then it kind of leaks back to baseline over time. So that's given by these terms. Neurons that are sensitive to odors are supplied with a current that is odor specific and activates specific odors in specific proportions. And the voltages then run through this term here, which reflects the connectome of the fly. So here, this is like for neuron I, we're going to sum over all other neurons J, the connectivity from J to I, and the amount of voltage or the amount of current that neuron I gets is going to be proportional to the amount of current coming out of neuron J. So this is a pretty simple model of exchange of currents and therefore voltages between neurons according to the wiring diagram that comes out of the connectome. So raise your hand if you know what a connectome is, and do I need to explain that? Raise your hand if you definitely want me to explain it. Okay, great. Okay, I'm happy to. Yes. So the connectome is a data object we get by taking the brain, soaking it in plastic and staining it with metal so that we can image it with electron microscopy. And then it is sliced super thin, and you get this sort of nanometer scale image of the brain over something like 3,000 slices. And then with a lot of machine learning tools, you can go back and segment that and find where a neuron sort of flowed across those images, and you can see its synapses where it connects to other neurons. So the data object is an n by n matrix where n is the number of neurons and the entry is the number of chemical synapses between those neurons. So it's like a roadmap, a detailed roadmap of the brain. Now, it comes from a single individual, right? So this is a whole nother thing, and the lab is working on projects to say, like, if we have an individual's connectome, but we have activity recording from another individual, can we update the specific weights of the one connectome based on the activity patterns of the other? But separate talk. Okay, so what's marked in blue in this equation are parameters that are given from experiments and from the literature. All of those weights between all of the neurons are measured in the connectome. And if we assume kind of generic physiological properties of the neurons, then things like the membrane capacitance, the resting membrane potential, the resistance of the membrane, and the amount of current that is injected into neurons when we give them odors are all things that people have previously measured. So we can potentially just plug in those parameter values and run this dynamical system. If you do that, it doesn't work straight out of the box, but we introduce a single free parameter, which is how responsive is a downstream neuron to the activity of an upstream neuron in general. So that's given by this A parameter here. We can make it cell type specific, so it's got a subscript which is for the cell type CI. And this is that's
on a per neuron basis across all incoming connections. So it's sort of a cell specific excitability.
It can either be just a single value for every cell or a value for every cell of a cell type. And a cell type could be projection neurons or ORNs or lateral neurons.
But I mean, it's not a CIJ, it's just a CI.
That's right.
It's not weighting the individual.
Yes, it's the sensitivity of the downstream neuron to activity of all its upstream partners put together. How much of a change in voltage does that downstream neuron get for the amount of current coming out of the upstream neurons? Yeah. And there's physiological interpretations for this, right? It has real biological units, and you would correspond it to the kinds and abundances of channels that are in that downstream cell's membrane. Okay, so that's what the matrix looks like. If we kind of rearrange it to look more networky and useful, this is these are the olfactory receptor neurons. These are the projection neurons that I was telling you about. Oh, sorry, these are the projection neurons. These are other cells in this circuit. We can run this. There's a term I didn't include, which is when the neuron hits its firing potential, it does an action potential. That's actually when you get the release of current out of that neuron. But it's a spiking model. And so you can play back the voltage and it looks like this. And this looks like reasonable spiking trace for, in this case, a projection neuron.
So Ben, maybe you mentioned it when I was away. These are point neurons? So no,
they're point neurons, yeah. You could imagine doing your cable version, yeah. Well, but the connectome could be great for this, right? But it has all that data. We've got all those kind of cable cross sections for every neuron, yeah.
Do you think going back one synapse is enough to explain the data?
In terms of the inputs onto each neuron? Well, so, I mean, the dynamics are recurrent, right? So the sort of second order inputs do show up in in the dynamics of each neuron indirectly through the immediate upstream one. I mean, this is the sort of canonical understanding for how chemical synapses work, which is that, you know, neuron J has a chemical synapse onto I, and if neuron K doesn't have a chemical synapse onto I, then its effect via chemical synapses at least would have to be indirect. Yeah.
So the couple of time course,
The time course.
you are recording data on the computer to the frequency deep reverse time each time. You added the stimulation and connectivity.
Can we, sorry, say it again?
Time course.
For the time course. So are you asking about the time course of the dynamics or? Yeah, so in this case, we're simulating like 0.4 seconds of dynamics. Neurons can fire sort of up to about 100 hertz. And you can kind of see the relaxation time of the voltage there is sort of millisecond or tens of millisecond timescales. Is that what you're asking?
I'm asking for the connectivity.
Yeah. The timescale of the connectivity.
Anatomically, each portion. Because sometimes varying the neuron is a certain population, not all population.
Yes, yeah.
Yeah, that's what I'm looking for a reason.
I see.
Yeah.
Like, do we see sort of a certain timescale of activity
Yeah, for a
moving
reason.
through different parts of the network?
Yeah.
I would say for this circuit, the timescale of the activity is dominated by the timescale of the inputs coming into ORNs. This is not like a highly recursive part of the network. And if we were doing the whole brain and allowing it to kind of free run without the constraint of inputs, you would see dynamics in the sort of tens of milliseconds out to seconds these days. Now, I think we don't know what the actual like free running timescale is because you can't really turn off stimulus input to a brain. Yeah, I hope that's an answer. Yeah. Okay, I'm sort of out of time, aren't I?
I've been asking questions, so keep going.
Okay, I'll try to go a little bit faster, and hopefully it can show you a little bit of the full picture. All right, so the approach at this point was we have our leaky integrate and fire model for the voltage, and there's a ton of fixed parameters in that, all of them except for that sensitivity parameter I told you about. So what we do is like we integrate this, we get predicted voltage dynamics per neuron, and we compare them to known electrophysiology recordings, mostly from our colleague Rachel Wilson, who's at Harvard Medical School. And then we just adjust that free parameter until the predicted dynamics look something like the measured dynamics. And we're assessing the average firing rate of neurons by class. We're assessing certain spectral qualities like how sparse or how white is the representation of odor information in different layers of the olfactory system. And just by changing that one value, we can get things to kind of agree. So this is a picture from the paper. You can see like ORN versus PN response curves, like transfer function curves here. This is for the literature, that's for the model. These are firing rate distributions for the literature and the model. And then this is that sort of PC representation of sparseness of odor coding for the data in the model. And they can kind of qualitatively agree, and we can say, looks good. Now let's sort of do our in silico experiment. And I won't go into too much of this to say, other than like, we can play back different developmental scenarios. We can construct that wiring diagram in ways that we think might reflect the idiosyncrasy of developmental processes, and then ask how does that change odor coding. So what you're seeing here are kind of individual, randomly redeveloped connectomes based on bootstrap shuffling of the real connectome. And then we can do this, skip. And the point is that when we add developmental variation to ORNs, we pattern the variation across individuals along the principal components in the same way that we measured the principal components of the like individuals by 60 odor glomeruli response values in these kind of glomerular clusters that look like the real data. But when we add developmental variation to local neurons or projection neurons, that's not shown, or just look at the principal components under Poisson timing of spikes, you don't see that glomerular organization. And so to us, this is pointing to developmental variation in ORNs as potentially the explanation for this glomerular scale patterning of variation in the calcium responses, which correlates to behavior. So now we're sort of able to make a prediction about which cell type it is specifically, where developmental variation produces the spectrum of responses across individuals. Okay, yeah.
I know we don't have a lot of time, but I don't think you quite said how you constrain that variation that you introduced in simulations. Because if it's completely unconstrained, then all hell will break loose.
You mean the business happening here?
Yeah. So you
Yeah.
presumably have some parameters for what to change and how much to change.
Sorry, the slides are laggy. Basically, we make buckets of what we consider cell types. And if you make the assumption that every individual cell you see within a cell type represents an independent sample from a developmental process, then we do bootstrapping within those.
Basically sample from some distribution that you create with some local.
Yeah, and it's all done, well, it's not done with like parameterized models. It's just done with the existing connectome.
Right, yeah.
And so so it's like buckets of neurons. And when we say we're going to make a new individual, we draw with replacement from that list of neurons in each of those cell types. And then now we have a new list of cell types by cell types. And then we just look up from the connectome what their synapse weight is. So you can see that sort of being applied. This should be animating. Well, this one animates. So you can see that it's roughly the same each time, but it differs in detail.
So that's development and not other stuff?
We're assuming it's development. And it'd be a development broadly construed that includes like all the homeostasis that happens in adults and so on. Yeah.
It's not the whole variability that you described elsewhere, just even.
Yeah. So in flies now, we do have multiple connectomes. There's the original hemibrain, there's a FAFB one, and then there's a couple recent ones that are whole CNS that aren't quite as annotated in as much detail. And it seems broadly consistent with that. Like, obviously, I don't think of individual cells as independent samples from identical distributions. But I think it's a good starting point, and it did give us an interpretable hypothesis with respect to the calcium variation. Okay, I'm way over, so I'm gonna skip all the philosophy. You can ask me about that. Okay, basically, because I want to show you like just the idea of bringing Bayesian inference into this. Instead of like taking that one free parameter, which was the sensitivity, and just kind of tweaking it by hand until we were able to match the experimental data, we actually want to do Bayesian inference on that parameter. So the workflow is now going to be that we have this dynamical model that has a zillion measured parameters coming from electrophysiology in the connectome. That's the same. And then there's now going to be parameters that we're explicitly doing inference on, and one of those could be that cell type specific parameter. So we're going to plug those in, do numerical integration on the dynamics, produce firing rates, which will run through a likelihood now to produce a posterior over those inferred parameters. And we can do this for different biophysical scenarios. So this is the leaky integrate and fire model with a global sensitivity parameter for all cell types. That's what I showed you. We can do it in a cell type specific way. We can do global sensitivity plus gap junctions, which we can approximate by the contact area that we infer between the neurons from the connectome. So we can specify different biophysics for what's happening at each neuron, and then still bring to bear all of these data points from the connectome. And so in all of these cases, the number of inferred parameters is small. It's like 1 or 3 or 2 in this case. And instead of that workflow where we just kind of eyeball it and say it looks good, we integrate, get voltage dynamics, run it through a likelihood of the experimental data given these rates, apply a prior, and update posteriors from this. And this is computationally kind of gnarly. So running enough simulation of the brain, at least on our cluster, maybe Janelia can do better. This takes 45 minutes per likelihood calculation. So you can't bring to bear the Bayes tools you're used to, like Hamiltonian sampling or Metropolis Hastings. Those require tens of thousands of steps, and tens of thousands of 45-minute steps is not acceptable. So we just do because we have these low dimensionality parameter spaces, we just do a grid search over those parameter spaces and calculate the posterior that way. And so here's this one-dimensional grid search over that global sensitivity. I don't have too much of a story on this yet, other than to say it's working and show you a couple places where the Bayesian inference is showing us some biology. So here's one. This is in a 3-parameter model where there are cell type specific sensitivities for local neurons, excitatory local neurons, inhibitory local neurons, and projection neurons. And these are the sort of marginalized posteriors of that 3-dimensional posterior along 2 dimensions or along 1 dimension. And what you can see in this heat map here is there's this diagonal band. Do you see the diagonal band here in the posterior? What that corresponds to is some kind of excitation inhibition balance that needs to be there in the sensitivity of excitatory local neurons and inhibitory local neurons for the circuit to account for the data that are in the electrophysiology. And here's another example of that in a gap junction and global sensitivity model. And if you look at the position of the posterior for the strength of gap junctions, it's non-zero. And this is in the antenna lobe. And we know that there are, in fact, gap junctions in the circuit. So we're sort of seeing some agreement now between the inference we might do over biophysical parameters coming out of this, aligning somewhat to the real known details of the system. But I think what's exciting about this is dynamical models of connectomes are sandboxes for experimentally intractable hypothesis testing. We can do Bayesian inference on the parameters of compact biophysical models. And the philosophy was like, can we have an F equals MA for neuroscience? I think maybe we already have it. I think it's things like leaky, integrate, and fire, and Hodgkin-Huxley, and so on. These are the rules that work at the local scale that potentially explain the global phenomenon. This is technically challenging and doable, and there's some hints that this approach makes predictions that are borne out by experiments. All right, thank you.